for this reaction has not worked at all. So what could have gone wrong?
I'm
Dr. Sayanta?
Good evening. I'm Dr. Shanta from.
Let's start. So, please. Yes, sir. It's visible. Full screen, yes.
So good evening, everyone. Me and Gaurav will take you through a few molecular techniques via few cases. So let's initially we'll see what is molecular biology. It is a branch of biology that deals with structure and function of macromolecules that include proteins and nucleic acid. These utilize nucleic acid like DNA, RNA, as well as proteins to understand molecular biology at molecular level. So in the molecular biology, we try to understand about human genome, which actually comprises of 22 autosome pair with 2 sex chromosomes. And in these chromosomes, we have actually multiple genes that are there. We have 30,000 to 40,000 genes that are there, which have actually 3 billion base pairs that are there. So if we look at a cell that has a nucleus, it has these chromosomes have multiple genes as well as inter areas that are intergenic regions are there. In the genes also we have coding areas that are exons and non-coding areas that are in form of introns. And these whole sequence, they are in form of nucleotides that are ATGC that are there. So if you look at the human genome, actually most of the region is non-coding, and only 1% of the whole genome is actually coding genome, or that actually makes sense. So if we look at the central dogma of molecular biology, we have the starting material where we have DNA, which gets transcribed to RNA, messenger RNA, which carries the message through translation and formation of protein in the cytoplasm. So this is the central dogma of molecular biology, where DNA gets transcribed to RNA, RNA gets translated to a protein, which gives a functional meaning. So the central part of a molecular technique, to any molecular technique, is the polymerase chain reaction. Polymerase chain reaction was discovered by whom? So can Deva answer this question? So does anyone? Yeah. Okay. Anyone else who knows who this person is? So Carl Mullis was a person who invented PCR, and he was awarded the Nobel Prize also in 1993. So Sayanta, can you tell me what happens in a PCR reaction? What are the components and what exactly is happening in a PCR reaction?
In a PCR reaction, we what we do, a specific sequence, we amplify it.
Correct.
First, through a primer, we anneal 3' region, then we amplify it, and then the cycle repeats. So we get many amplified sequences.
Okay, so so in that, if we see what are the components that we will require for PCR, we would need a template, as you said, DNA will be required, right? Or RNA, whatever is there, that will be required. And then you need primers so that you can identify which areas to amplify. And then you need nucleotides that are there, and you need DNA polymerase that should help you for addition of these nucleotides. And you also need a buffer solution. So if you look at schematic diagram of what you see, so what happens? You have a double-stranded DNA structure, where primers are supposed to go and bind. Now, before primers have to bind, this double-stranded DNA has to break into a single-stranded structure. So basically, denaturation has to happen, where at a temperature more than 90 degrees, at around 95 degrees, your 2 DNA strand will get separated. Once they have got separated, a temperature of 60 degrees around, this may vary according to primer. It is around 58 to 65 depending on your annealing temperature of your primer. It may vary, but approximately at 60 degrees, your primers will go and bind to the site of interest. And then in the presence of DNA polymerase, extension will happen at a little higher temperature of 72 degrees Celsius, where your DNA polymerase will get activated and it will be adding the nucleotides in both directions. So what was initially at the end of one cycle of PCR, we will get 2 copies of DNA, double-stranded DNA, we will get 2 copies. So similarly, this is repeated for multiple cycles to get millions of copies of DNA. So basically, to visualize what is there, any change that is happening, what we are doing is we are amplifying a sequence. So this is called PCR, and this is one of the central technique of our molecular biology, where based on which our techniques are based. So now once we have amplified the product, how can we visualize these products? So according to the visualization process, our PCR technique may differ. So basically, there are various kinds of PCR, and so in today's example, I had given example of RT-PCR, reverse transcriptase polymerase chain reaction, in a case. So let's go through that case and see what is there. So can Tapan read the history of this case?
So case 1, a 55-year female with gagging sensation in the abdomen. On examination, the spleen was palpable 10 cm below the left costal margin.
Correct. So what do you think till now? What possibility will you think with this history?
Ma'am, a gagging sensation of the abdomen with a splenomegaly, we will try to rule out the causes of individual splenomegaly. So we'll take further histories to rule out any causes that can be either hepatic, or they can be congestive splenomegaly, they can be hematological disorders which can cause splenomegaly.
Now we go to the peripheral smear, where you are getting this kind of morphology. So what is the prominent finding here?
Ma'am, there is hyperleukocytosis with a myeloid predominance, showing predominantly neutrophils as well as immature myeloid series, myelocyte, metamyelocytes. We can also see few eosinophils, few dysplastic forms are also noted. Few blast-like cells are also seen.
You mean this would be blast-like cell?
Yes.
Shweta, can you tell me?
I'm going to raise a pointer. Yes, sir.
Any basophil we can show?
Yes, so this you mean is blast-like cell, where with a prominent neutralized scant amount of cytoplasm. Here we are seeing this would be, I think, pro-myelo, because neutralized is kind of visible. Then here we have myelocyte. These are eosinophil. This is another blast that we are having, and this should be a basophil. Here we can see coarse granule that is there. They are better basophil in bone marrow picture that we can go. So with these findings that we have leukocytosis, shift to left, basophilia. Here we cannot call myeloid preponderance. Why can't we say in peripheral blood myeloid preponderance word?
Because we cannot say whether the other series has been suppressed in the proliferating organs, and the RBC's picture as well as the appears normal in the smear, I'll leave it also.
Correct. So actually, in bone marrow, you are supposed to have normal erythroid precursors, but in peripheral blood, you're not supposed to have erythroid precursors, right? So we cannot call that use the term myeloid preponderance in peripheral blood finding. What we can say is that there's leukocytosis, there's shift to left, and there's basophilia. Now, what would you want to do next, Vishwabandhu?
Advice for bone marrow aspirate.
Bone marrow aspirate. So do you really need bone marrow aspirate? Like peripheral blood is there. Do you really need bone marrow aspirate in such cases?
I think we should.
Sorry?
I think we should need to rule out
Yes. Rule out what?
the cause like CML.
Okay. See, for example, here I went ahead and performed FISH in this. Okay, so we have green signal that is coming from the BCR, and we have red signal that is coming from ABL. Okay, we have these yellow signal that are coming from BCR ABL fusion. Like we have two yellow signal here and one red, one green separate signal here. So this is BCR ABL fusion. Now I have got this BCR ABL fusion here. Then do I really need to go ahead for bone marrow? Because CML we have confirmed, and we had 2% blast suppose in the peripheral blood. So do we really need to do bone marrow?
We can classify the phase whether it is chronic phase or blast crisis phase.
Correct. Correct.
Identifying the number of blasts. So
Correct.
this can help in the prognosis. Also.
Correct. So we have a diagnosis where we know there is reciprocal translocation of BCR ABL has happened, where because of formation of BCR ABL fusion, we will have constitutive activation of BCR ABL gene, and that will lead to formation of chronic myeloid leukemia. So yet we went ahead and performed bone marrow because it will help us to distinguish between chronic phase and blast crisis, as well as cytogenetics is usually possible only in bone marrow because metaphase we get in bone marrow and we don't get that in peripheral blood. So apart from that, biopsy will also help us to look at fibrosis, and biopsy also may identify nests of blast. So here in the bone marrow aspirate, what is the finding here? Preeti, would you like to describe?
Yes, ma'am. The bone marrow aspect of the particle, we are able to see it is a cellular particle. Ma'am.
So if this age was 50 something, so cellular means we want to commit to hypercellular, hypocellular,
Hypercellular, it is a hypercellular aspect.
correct? Then can you see these things? Yeah, increased.
Megakaryocytes.
Correct.
Megakaryocytes are also there. But increase in myeloid cells. Ma'am.
Correct. So now here we can use the term myeloid preponderance. And what is this cell here, Preeti?
It is a basophil.
Yes. Okay. So we have myelocyte, metamyelo, we have eosinophil that are there. We have basophil. So shift to left. And so what is this cell?
Nucleated RBC.
Correct. So we have basophilia. We have nucleated. We have very few nucleated RBC. Most of them are myeloid cells that we can see. And blasts were also less than 5% in marrow also. So we have a diagnosis where we have done bone marrow aspirate, where we are sticking to chronic myeloid leukemia chronic phase, and we have done FISH. Now do we want to do anything else? Deva.
Ma'am, we can go for PCR.
Okay. So how will PCR help us now? Because in PCR also we'll come to know that it is BCR ABL, right? How is PCR helping us?
Ma'am, we can go for quantitative PCR to see the quantity of the amount of BCR ABL fusion is there.
So suppose I get amount of BCR ABL as 100, or if I'm getting a 70 ISNCN, let's say that way. So how is it different? Like how is it making a difference for a diagnostic patient?
Ma'am, it can help in monitoring the therapy in future, not exactly for diagnosis.
Okay, okay. So basically for monitoring of therapy, if it will help, we just want to know what kind of BCR ABL transcript is there. So at baseline, are we getting P210, P190, which transcript we are getting? That becomes important for PCR. Another thing that we also do is conventional karyotyping. So in conventional karyotyping, what is the finding here? Sayanta, do you want to tell?
In this, the Q arm of 22nd chromosome is shortened, whereas the Q arm of 9th chromosome is of a larger length, and this denotes a translocation between 9 and 22 chromosome.
Okay, so for which of the chromosome do I call Philadelphia chromosome? Chromosome 9 or chromosome 22?
22.
Okay, so the shortened chromosome 22 is called Philadelphia chromosome, correct? So here we had translocation 922 that was there in the karyogram. So we went ahead and performed RT-PCR, and it is mandatory to identify the type of transcript here. So what if we did a quantitative kind of PCR at the diagnostic time point? This won't really help us because in case it comes positive, well and good, but if it comes negative, it will not help us because can be variant transcripts that may be happening. Like the common translocation of BCR ABL happens between the exon 14 of BCR and exon 2 of ABL. But sometimes there may be exon 3 of ABL involved instead of exon 2. And atypical BCR breakpoint also may happen, where breakpoint in exon 6, exon 8, exon 19 may happen. So these cases will not be identified by your quantitative PCR. So in qualitative PCR, whereas you can do a multiplex PCR, where you are amplifying a control area in the BCR gene. Here in the control area, you can see a product of 808 base pair. And in case you have E13A2, that is breakpoint in exon 13 of BCR, and exon 2 of ABL gene, you will get a product of 310 base pair. E14A2, you will get a product of 385 base pair. And for your E1A2, that is P190 kind of product, that is your one which we see commonly in BLL, you will get a product of 481 base pair. So in the same reaction, you are getting products of different base pair, and you will be able to identify what transcript is there. You can also go for variant PCR of BCR ABL, where you can amplify the area of exon 3 also and exon 6 and 8 of your BCR ABL. So here, what we are doing is we are using multiple primers, and whichever is present will amplify. So we can identify whichever transcript is there easily in case of RT-PCR at baseline. But in real-time PCR, what happens is, where you want to quantify, you will be able to amplify only one small area. So for real-time PCR, for the major transcript, that is E13A2 and E14A2 is a different type compared to what is there for E1A2, that is P190 transcript. So for example, a BLL case that comes for monitoring, for that the real-time PCR that we'll be putting will be different from compared to the case that has classical E14A2 transcript that will be coming for follow-up. So that is why we have to identify which transcript was present at baseline. Gaurav, do you want to add anything? Yeah. So if we look at the BCR ABL, the commonest transcript that we get is E14A2 in 63% cases, and E13A2 is there in 20% cases. But even in CML, we can get E1A2 in 1% cases. So we have to identify what transcript was there in baseline, so that exactly the same transcript we can identify in real-time PCR during monitoring time point. Here, if we look at this PCR, so Tapan, do you want to interpret? This is our patient. Okay, so we are getting two bands. One is at 808 base pair. First of all, on the right, we have molecular base pair. So this is 100 base pair, this is 200, this is 100, 200, 300, 400, it goes this way. And this is 1000 base pair ladder. So now compared to this, if we try to compare, so this is 100, 200, 300. So this band is near around 300 base pair. And this band is nearing around 400 and this band is nearing around 500. So similarly, this upper band is nearing around 800 base pair. So in our patient, we are getting two bands. One is at 800 around base pair and other one is nearing around 400 base pair. So do you want to interpret, Tapan, what are we seeing in this case?
Ma'am, in our case, the patient's band is resembling the 385 base pair band, that is E14A2.
Correct.
Most likely on the transcript, we are dealing with is the E14A2.
Okay. So in this patient, we are having E14A2. Now here, what is this band that we are seeing? That 800 base pair band, what is it?
Oh.
So if I go back to the example, here I have shown that we are also putting primer for the control region. That will give us a band of 800 around base pair. So this we will be seeing in every case. Okay. So now suppose this first case that is there, it is showing us only band of 808 base pair. So, but there is no other band that is there. So what will you consider this case as?
Control. And control.
For prime.
So Preeti, you were saying something? So the control primer or control region has worked, but there is no other band. That means this case is negative for PCR label. Okay. But now at the same time, if I'm looking at this link, if I tell you that I had put a PCR for this also, but there is no band at all, can I call this negative?
When the reaction has then not occurred because the control band is not there.
Correct. So our internal control for this reaction has not worked at all. So what could have gone wrong?
Ma'am, there can be when the DNA might have degraded, and there can be the temperature differences. Any link has not occurred properly. There can be magnesium content alteration in the reaction mixture. The primers might not be working, and so we have to troubleshoot all of the following and then to realize that.
Okay. So now in this situation, I would say that since in other, because you put multiple PCRs at the same time, right? So since these have been put together, I don't think this would be a problem with magnesium concentration or other thing, because if it was a problem, we would not see anything in the entire gel. But here other samples have worked. That is sample related issue here, not the reagents related issue. Okay. Okay. Now suppose if I saw a band in NTC, what do we mean by NTC here?
Negative template. No template control.
No template control. Suppose if I had seen a band here, that means that would indicate that there is contamination of the master mix also. So I cannot interpret my complete reaction. So basically every time a PCR goes, because it is, thing of microliters that is going, contamination and these things are a lot of issue. So we do have to have our positive controls. We have to have no template controls in every reaction. Okay. So we have a PCR ABL, transcript. So now you were telling me that the DNA might have been degraded, but was DNA the starting material for RT-PCR? What is the full form of RT-PCR?
Reverse transcriptase polymerase chain reaction.
Yes. So in reverse transcriptase polymerase chain reaction, what is the starting material? DNA or RNA?
RNA. messenger.
Correct. So why are we using RNA and not DNA for identification of BCR label or any translocation for that matter? Can we use DNA for RT-PCR?
This one can be done.
It can be done. So what will be the difference? Like if I use DNA versus RNA, what will the what problem can I face?
And non-functional mRNA suggests that the DNA is transcribing, so that what then it can.
Not really. So basically, what happens?
So we can detect BCR label translocation in both DNA and RNA.
Correct,
No, it can be in both. So why RNA? Why not DNA? That's the question.
So let me show you. So basically, we were discussing that we will have coding and non-coding region. And in case of, if we deal with this example here, we have 3 exons and 2 introns here, right? So this blue area is our exon and this pale areas are our intron. So if you look at the coding region, or exon 1 is 200 base pair and intron 1 is 16,000 base pair. Again, exon 2 is 200 base pair, intron 2 is 15,000 base pair, and exon 3 is 300 base pair. If I wish to amplify this area, like I just want to amplify the coding area also, I just want to know if this is getting amplified or not. But if I do at DNA level, I will get a product of 15,500 base pair. Okay, so just now I showed you a gel picture where I had used DNA ladder up to 1000 base pair. For 15,500 base pair, the gel handling will be very tough. Like it is like very tedious to do such kind of PCR where you are getting such big products. But at the same time, if I'm doing RNA level product, I will get a product of only 500 base pair because it is giving me only coding area. And usually translocations, one of the gene will go and fuse with other, and it becomes very easy if you are using a cDNA, RNA and cDNA rather than using DNA. So in cases when we want to look at gene expression, when we want to look at translocation, we go ahead for RNA based test in such scenarios. Okay. Was diagnosed as CML and was started on tyrosine kinase inhibitor. So what is the mechanism of action of tyrosine kinase inhibitor? Can anyone tell? So Deva.
Tyrosine kinase actually helps in signaling and helps in proliferation of cells. So basically, the inhibitors like imatinib, dasatinib binds to the tyrosine kinase and stops the signaling pathway.
Okay, so tyrosine kinase inhibitor or imatinib binds actually to the ATP binding site of this tyrosine kinase. So basically, it will not allow the downstream pathway to act. So in 2001, FDA had approved imatinib, and before imatinib era, CML wasn't that great a disease. Nowadays in OPDs, when patient comes with CML first time when the counseling happens, the hematologist says the patient, you have got a blood cancer, but it is a good type of blood cancer, because the survival rate have really improved post tyrosine kinase inhibitor with the survival reaching up to 80% in 10-year survival reaching up to 80% also in such patients. So this was published in Time magazine, where it was called a magic bullet drug, imatinib, when it was approved for the first time. So now, once you start giving imatinib to these patients, how would you monitor these patients, Sayanda?
Monitoring for imatinib therapy, there are 3 facets of this. That is hematologic remission, physiologic remission, and molecular remission.
Correct.
So for hematologic remission, we have to check the PBS and bone marrow. For
So I'll agree with peripheral blood, but not bone marrow because okay. Then next,
cytogenetic remission. For that, we have to do fish.
Correct.
And for molecular remission, there are levels there. That is deep stable molecular remission,
Okay, you're going to levels, but what is the technique by which we will check BCR-ABL in the
We
molecular?
have to do the quantitative PCR for that.
Quantitative PCR or real-time PCR?
Real-time
So,
PCR.
yeah, okay. So if you look at the sensitivity of these different techniques, so you will be able to decipher using real-time PCR up to sensitivity of 0.001 also. So this is a very sensitive method of doing testing these patients during follow-up time point. So after 9 months, this patient, a real-time PCR was performed. So how is this real-time different from what we had done at the diagnostic time point? What in the materials we had used primers, we had used VNTPs, we had used buffer that was containing magnesium and DNA polymerase. So what additional thing is there during the time of real-time PCR? What was not there at RT-PCR? Vishnu,
Maybe the quantity of the protein this decrease.
so when we saw in the gel picture, we couldn't quantify, no, this would be 10%, 5% or so like that, right? Now in real-time PCR, you get report of 0.01 also. So for that, is the method different from what was done earlier? RT-PCR, was it different? Tapan, would you like to answer this?
Some fluorescence probes are used
Correct.
for real-time PCR. So
Correct.
they are based on the fluorescence resonance energy transfer. So the probes are linked with a fluorescence molecule as well as a quencher. So
Correct.
they bind with the DNA part, which is the target gene. Then whenever DNA polymerase starts working, it disrupts the binding of the quencher and the fluorescence molecule, so that when light emits, then the fluorescence starts, and it can be detected based on the number of application modes.
Very, very beautiful description. So basically, you've done the part that I was supposed to do. So here, the probe binds, in addition to the forward and reverse primer, we have a probe that is bound to fluorophore at the 5' end and quencher at 3' end. And when it is intact, it doesn't emit any fluorescence. But as soon as there will be amplification occurring, this fluorophore will get separated from the quencher dye, and then you will get a light emission that can be detected. So what you get is in form of these curves, where you can detect in real time how the signal is, at what point the signal starts. For example, this became very famous during the time of COVID, CT value, COVID PCR, CT is this, CT is this. So basically, the lower the CT value, that means earlier the amplification has happened, higher the viral load is there. Okay, so what happens? We run standards of known concentration and plot this CT value. And once we run patient sample, and whatever CT value we get, we can extrapolate that this is the quantity of. Okay, so here this patient was at 42 months. The BCR-ABL copy number rose significantly, and we got NCN or normalized copy number of 5.6. Okay, so now what would you like to do? This is rising trend of BCR-ABL. We had reached 0.01 level, but subsequently on monitoring, at 42 months, we got increasing trend of BCR-ABL. So what would you do now, Dr. Preeti?
No, maybe the patient is in remission.
So a patient is not in remission. So is there any other molecular technique you can do to find out why this is happening? Anyone else? So we go ahead and do kinase domain mutation testing. Has anyone heard of this? So have you heard of mutation T315I? In when you read this, what are the CML criteria? Have you read about the mutation T315I? So once a patient receives imatinib, most of the patients respond to this. Okay, but some of these patients, they develop secondary resistance, and secondary resistance happens when there is a mutation occurring in the kinase domain. So kinase domain was a domain in the ABL region that was where your imatinib was supposed to go and bind. But if there is a mutation, it will not allow imatinib to do its action. Okay, so T315I is one of such notorious mutation which is known for such such a feature. So here, what can you see? You can see nucleotide data there, C, C, G, A. So what do we call such kind of picture? Anyone?
Sanger sequencing.
Correct. So Sanger sequencing, chromatogram. So this is an output of Sanger sequencing, which is in form of chromatogram, where we can see the nucleotides here. So A, T, C. So here, what are we seeing? We are seeing
Double
two
P.
nucleotides. Correct. So here we have a heterozygous kind of mutation. We can see, or there are two peaks. Okay. So what was supposed to be A, C, T. So A is green color, C is this blue color, and T is red color. Now we have this has changed to ATT. So ACT coded for threonine. However, ATT codes for isoleucine. So what happens now? At 315 amino acid position, what was coding for threonine now has become isoleucine. So we can say that this patient has mutation T315I using the Sanger sequencing chromatogram, where we found the sequence. So what happens? What is the principle of Sanger sequencing? So apart from the primer or DNA template that we had and DNTPs, we also have DDNTP. Okay, so which is dideoxynucleotides. So they are bound to fluorochromes. So as soon as these bind to a template, chain termination happens. But they are bound to fluorochrome. So once they pass through capillary electrophoresis, you can decipher the sequence. So let me explain it to you with a better example, or a better slide actually. So our DNTPs, they have OH, hydroxyl, at their 3' end. However, if you have this DDNTPs are modified in such a way that instead of hydroxyl group, they have hydrogen in their 3' end. So once this hydrogen occurs, chain termination will happen. That means elongation will not happen at this end. And this DDNTP, we have bounded to a fluorochrome. Okay, so what will happen if your when the DNTP is added, your phosphodiester bond will get formed. However, wherever DDNTP is added, your phosphodiester bond cannot form, and there will be chain termination that will be happening in Sanger sequencing. So by this, what will happen? You will have fragments of various lengths which will have terminated at different lengths, which when you pass through capillary electrophoresis, you can identify that what nucleotide has got incorporated when, and they will be serially displayed in form of chromatogram. This is how you can decipher sequence in Sanger sequencing. Okay. So in this case, where in a CML case at diagnostic time point, we use RT-PCR. For monitoring, we used a real-time PCR. And when this patient had increasing trend of BCR-ABL, we went ahead and did kinase domain mutation testing by Sanger sequencing. So this was from case 1. If any of you have any questions, we can address that. Otherwise, I'll pass the baton to Dr. Gaurav for next case and explaining the next.
So I think I just want to focus a bit on what Shweta said. So as a pathologist in this case, to see at what are the steps that you could impact patient outcome, right? So you diagnose that this patient had a molecular abnormality which is very much responsible to a particular therapy, right? So the clinician could give that therapy and the patient responded. Then you could monitor the patient using another technique, which is qPCR in this case. And then finally, when the patient stops responding and the disease starts coming back, you can do another test, which is standard sequencing in this case, to determine that what may be the cause of this change in response. So based on that standard sequencing result, if you find specific mutation, so they could change the TKI, they could consider transplants, all that. So this is the kind of different levels that molecular technique do impact in today's date patient management. So if you have any question regarding this case, we can take it right now. Let me see if there's anything in the chat box.
So there is nothing in the chat box regarding this case. There's one question. Please explain the transcripts in CML.
Yeah,
Correct.
Shweta, maybe.
Yeah. So let me share it again. Yeah. So now, when BCR ABL fusion happens, you may have breakpoint in different regions in BCR and different regions in ABL. So if you have major transcript are P210 transcript, that is breakpoint occurring in your exon 14 of BCR. So this is exon 1 and this is exon 13. So this one is example of exon 13 of BCR where breakpoint has happened, and exon 2 of ABL breakpoint has happened. So this fusion is happening in E13A2, it is called. And if the breakpoint is happening in exon 14, you will have E14A2. For both of them, we call the transcript as P210 because exon 14 and exon 13 are nearby and they can be monitored using the same real-time BCR. So basically, this is called the major transcript of BCR ABL, which is the majority component of BCR ABL fusion that happens in CML. Now, other transcript that E1A2 is where a breakpoint happens in exon 1 of BCR and A2 or exon 2 of ABL, and this happens commonly in BALL, but very rare in CML. Okay. So now in other variants may happen. So basically, you may have breakpoint in exon 6, exon 8, exon 19 of BCR, and these are there. But these are important for you to know that at baseline what transcript was there for you to monitor. So I just saw the question that will they be important for outcome. So I don't think they make a difference in outcome, but it is just that you want to know the type of transcript so that you can monitor them. So quantitative PCR is available for E13A2, E14A2, the major transcript, and P190, but it wouldn't be available for variant transcript. If you want to monitor variant transcript, you will have to do nested PCR. So if suppose a variant transcript patient, you are just doing quantitative PCR of, you will be won't be true. So this is important for the monitoring purpose that you should monitor the same transcript that you are detected at the diagnostic time point. That's all.
Yeah. Another, I think, curious implication of this is that sometimes we see cases where FISH is positive but PCR is negative. So that often happens because people will do very commonly the common transcripts like E13, E14A2, and E1A2, and will miss all these rarer transcripts. So they will say that it's not CML, it's PCR is negative, whereas your FISH is positive. So how do you then tally them together? So the fact is you have to always check what test exactly was done so that you know that how to interpret that test in a much more informed manner. So that's also again important. We will argue that more in the next case, that how molecular is very, very important in the manner that it's a specific test. We have to ask very specific question. And there's a question about additional cytogenetic.
Yeah, correct. So basically, I did not cover that because I was covering more of PCR and real-time PCR. However, additional cytogenetic abnormality or clonal cytogenetic evolution term is there that is being used in parent WHO as well as ICC. So basically, your presence of additional Philadelphia chromosome, isochromosome 17, or trisomy 19 that is there, and they are complex karyotypes. So these abnormalities are there that you have to list as clonal cytogenetic evolution that is present.
So, sir, if the major transcripts come negative, then shall we go forward and next gen sequencing or something, or do we?
So basically, most of the lab will have variant transcripts PCR that is there. So if that also is negative, let's go for RNA sequencing and see if it is something else.
Yeah, we will have some examples in the
Yes,
next case also, right? So no. Doesn't the additional cytokine changes do not change the monitoring strategy because that depends only on the transcript, right? So that's not on the additional cytokine numbers. It's just a mechanism of disease progression in CML, but the fundamental transcript will be there, right?
So we have two questions, I think, in the
This is something
yeah. So basically, it was said that in chromosome it was U that was labeled. So it is just the chromatogram, what it is reading that way. We have to know that what was there. So basically, we just interpret U as T for our purpose and not as U. It is depending on what software you're using. And I think Srinidhi has asked that based on length. Let me see. I don't know.
You have to stop the chain.
Okay, we have to stop the chain. So in standard sequencing, does the length of fragment correlate with the sequence of nucleotide?
No. It only there's a basically what you do is you start with a random mix of DNTPs and fluorescent tagged DNTPs, right?
Yes.
So whenever the fluorescent tagged DNTPs are incorporated randomly, in one cherry might incorporate the sequence number 4, in another cherry might get incorporated in sequence number 11. So that way, it doesn't at any time point that fluorescent level DNTP is introduced, the chain is terminated, and you have to then look at that thing. So length of the fragments only correlate with the moment that DNTP is being introduced.
And also the strategy here that is done is the DDNTP, lesser amount of DDNTP is used so that chain termination doesn't happen very frequently. So it should allow the length to go on. So we have not gone into detail because we are trying to touch upon the technique so that expose the students to the technique. And so I also have got a question that explain the concept of international standard. So I just give you the example that international standard is something like what we do for INR. When we calculate INR, we use like PT from different lab may vary. Okay, so we do use ISI, right? So but similarly here we have to use conversion factor that we compare with the standard so that be it lab in Mumbai or be it a lab in Punjab or be it a lab in Bihar, it should give the same value. Just like PT and INR, international scale is used in such a way. So to explain that concept, we will need a full a bigger length class. Let me touch it that way and you can read about it. You can always send your queries to email in case you don't understand.
Okay, so we had an excessive discussion. Nice. People are at least not asleep. That means okay. Right. So maybe we can take it. Who has been quiet so far? Vishwabandhu, maybe? Yes, Vishwabandhu, can you please discuss the history of this case? Vishwabandhu, yes.
Presented with fever and bleeding response. There is no history of comorbidities. And as is number CBC hemoglobin 5.2, total leukocyte count.
There's some, yeah.
and bone marrow person on 24/7/2020, and on flow cytometry AML CD34 positive, and then on molecular testing FLT3 ITD, so low allelic ratio.
Okay, so what is the interpretation of this, CBC? This is of course all in WHO unit, right? So maybe I should have mentioned the unit. That's all in WHO unit. So what is your interpretation? Is it normal? This now?
No, this is there is decrease in hemoglobin. That is pancytopenia. I think pancytopenia.
Correct. So there is a young female, young girl basically with pancytopenia, right? And the outside this patient came to us with an outside diagnosis of AML, which was CD34 positive. Okay. So we'll go to the plots now. So what is your impression of morphology? So peripheral blood, we could pick up some cells like these. I'll go to marrow. So these are the cells. How do you describe these cells?
These are the
First,
myeloblasts.
what is the severity of the particle? What do you think about this? For a 15-year-old girl,
The hypercellular.
looks like hypercellular, completely packed. The marrow is completely packed. Surely it's hypercellular. What about the cells? How do you describe them?
Look like blast. Might be myeloblast.
Okay.
Which are the high NC ratio with fine nuclear chromatin, and there is indented nuclear membrane with prominent multiple nuclei, and the cytoplasm is scanty. Scanty, moderate to scanty.
So I mean, okay, but there is area of cytoplasm I can see. Like for example, this here the cytoplasm seems to be quite abundant to me. Any other quality of cytoplasm that you could pick up here?
Here seems to have some granules in the cytoplasm.
It's not some granules. I would say it's packed with granules, right? There are a lot of granules. If you look at this peripheral blood picture, a lot of granules are there, no? So cytoplasm is packed with granules. Like you said, there is some indented nuclei here, right? So what is the morphology? What about this cell? What are these guys here?
Viruses.
Okay, only one.
Bundle of rods,
Multiple viruses.
What are they called?
agates.
So what do you think on morphology? What is your diagnosis? This diagnosis, of course, you have to make on morphology only, right? No other way.
Acute promyelocyte. We should have
Sorry,
promyelocytic leukemia.
acute promyelocytic leukemia, right?
Yes.
So what if I tell you this is normal promyelocyte? What is the problem? I'm arguing that these are normal promyelocyte. Okay, all these guys are normal promyelocyte. Even here you can see this is the normal promyelocyte. This has some rods, but it's again normal promyelocyte. How will you argue that this is not normal promyelocyte? Anyone, maybe Preeti or Deva?
Cytoplasm showing some blood.
Cytoplasm showing what?
Blood.
Indentation and bilobe are generally seen in abnormal promyelocyte, and Faggot cells are generally seen in abnormal. It's not in normal.
Sure, we were right. But also, I think one of the key point is that you can see a complete maturational block here, right? So all the cells that you're seeing, they are completely seems to be at a single maturational level. All of them are promyelocyte, right? So in case of leukemia reaction or stuff like that, usually you will not see like that. So there will be some maturation happening. They will not be monomorphic. All the cells are promyelocyte. This kind of feature will not be there. Having said that, there could be cases like this where the child has presented with pancytopenia and peripheral blood, especially you may have to hunt for cells like these. And even if you find 2 or 3 cells like this, you should be suspicious of the diagnosis. But generally speaking, FEML is a disease which is characterized by maturational block from biology perspective, and therefore you see this kind of monomorphic population. So that's also important to remember that we do not usually see any maturation. We see similar level of cells. So we did an cytochemical MPO, which is very helpful in these cases, and they were quite strong positive. So when I say strong positive, I mean that all of the promyelocytes, you can see they were nicely positive for MPO, so which is typically seen in this kind of cases. So then my question to you is, why is like typically these are known as Friday night or Saturday night leukemias, right? APML. You usually find them when you're on call, when you are just maybe going to start your weekend and you find a case like that and you have to see and make a diagnosis. So why is this diagnosis so important to make? Shanto?
Because it has a therapeutic implication. With retinoic acid, the prognosis is very good.
And if the therapy is not initiated, what is the problem? Maybe this has a therapy, okay. But why do we have to make the diagnosis then and there?
The patient's condition can worsen and it can cause DIC.
Right. So therefore, this question that what further investigation, the answer to that is, like you said, you must do coagulation test. These patients are prone to go into DIC, and therefore, and the fact that they are highly curable with specific therapy. So that is why the diagnosis needs to be made very, very fast. You know? So can you tell me maybe just a quick question to anyone? What is the first successful application of target therapy in hematopoietic neoplasm? Can anyone know? Like, does anyone know what is the first application of target therapy in hematopoietic neoplasm? Is it CML and hematite, like Shweta has discussed?
APML, I think in 1980s, the Chinese people started giving ATRA to the patients.
Very good. So the answer to that is APML, and we have ATRA. Even then we had arsenic trioxide as well, but ATRA was the first successful application of target therapy. Right. So then if you go to flow cytometry, so in this case the flow cytometry was done. So I now want some of you maybe who has not spoken. Preeti, maybe just interpret these 2 markers for me. So the red population is marked in, this is your target population, and the lymphocytes are in this thing you can see in sky blue or sky blue. Gray is your lymphocytes. Little bit of granulocyte there is in green, but the red is your target population. Okay. So tell me the expression of this population with respect to these 2 markers, CD34 and CD19. What do you think?
So the target population is positive for CD45, sir. So
Okay.
the
I'm asking
scatter
specifically for this plot. 34 and 19. I'm asking you to comment specifically on this plot. 34 and 19.
sir, most of the cells are so few cells are positive for 34, and the large population of cells are showing negative for 34, sir.
So here, just for your, just so that you can understand easily, these are the normal controls for 34, right? This black thing that you can see, these are normal myeloblasts. These are the normal control of B cells for 19. These are the B cells, right? So our population seems to go towards the 34 seems to reach to the level of this normal myeloblast. 19 seems to reach to the level of these normal B cells. So would you consider this as positive then?
Yes, sir. They're positive.
Typically we will, right? But what have you learned about the immunotherapy of APML? Are they typically 34 positive?
No.
Or
No,
negative?
sir. In APML, the HMATR 34 will be negative, sir. To the negative.
And what about 19? Should they be positive? These are not B cells, of course, right? They're myeloid cells. So should they be positive for 19?
No, sir. They will be negative for 19.
So what is the problem then? Are we dealing with an exceptionally rare case of APML, or is there a problem in our flow cytometry? Why do they seem to be positive? Does anyone know? Does anyone have an answer?
Mixed phenotype leukemia.
Sorry?
Sure,
Somebody
Could be mixed phenotype leukemia.
Oh wow, so you're thinking about mixed phenotypic acute leukemia, right? So this is probably I shouldn't talk about it today because this is not our topic of discussion today, but just because we're discussing APML. So there is something known as autofluorescence, okay, in the context of APML. So whenever you see this signal coming from flow, this is not CD34 to be honest. This is this fluorochrome which is conjugated with CD34, right? And the signal is coming actually from this fluorochrome, which in this case is PC5.5. In the case of 19 it is ACD, right? So what if the signal is coming just from this fluorochrome and it is not due to the attachment of antibody? Can that happen in life? So can there be some background fluorescence? The answer is yes. It's especially APMLs are very, very notorious for this. They do have this phenomenon known as autofluorescence. So for this, what we typically do is we do an unstained tube. So you can see that here we did an unstained tube and we just put PC5.5 and ACD and not the marker, right? There is no 34 and 19. it is only PC5.5 and ECD. And you can see that it's showing exactly the same pattern. So that means that this is just a background staining. This is just a benchmark. And only if it is crossing this limit, only then I can call it positive. So now if I put a gate based on this benchmark, it seems negative, right? Now it is not crossing the limit that was set based on your benchmarking. So I wanted to show this just to prove the point that in any test that you are doing, the benchmark is exceptionally important, right? You have to set your normal, otherwise you can't really interpret anything. So in this case, the normal is going up, the background is going up to this, and therefore I will not consider this as positive. This will be negative. So the outside lab made the same mistake. If you remember in the history, this was a AML 34 positive. It was called, but 34 actually is not positive. It is just a autofluorescence of the background fluorochrome. Okay, so we forget flow for now. Let's come to this patient. This is again fish. This is something Shweta showed you, so I'm not going to details. But in this case, this is again a fusion probe. You can see that this is the normal. There is a green for RARA and a red for PML, and they're fused here. Clearly, it shows a fusion happening. So therefore, PML RARA is positive. So again, we went for PCR. So again, the question just to rehearse what you learned. Maybe I can ask Deva. Will you do PCR or will you just be happy with the fish diagnosis of PML RARA? Deva, anyone?
So fish diagnosis should be enough for diagnosis.
Correct. So in that context, morphology is enough for starting therapy, right? So that is okay. But would you do PCR or not? And if yes, why? The default answer is no, right? If you don't like why do you need to do something else? Reason for it, right? So if yes, what is the reason for it? Tapan, maybe you can chip in.
Variant fusion, sir.
Of course, so we have understood the same concept in the CML, right? So the transcripts can be various. You can see here in PML, the breakpoint can be in different exons, like exon 3, 4, 5, 6, 7, anything can be broken. As in RARA, it can be either in exon 2 and exon 3. So based on that, various combination of these transcripts are possible, right? So you need to know the exact transcript that this patient has, just for the reason that you could monitor them properly, right? So just like CML, we will monitor these patients using real-time quantitative PCR. So for that, I want to know what is the transcript. So for this reason, we must know that what is the baseline transcript. The concept remains the same for CML and ATML in this regard. Why do you need to know the transcript? So just to rehearse, we know that ATML is a disease, one of the most we could say it's a poster boy of success in hematology neoplasm. What was a highly fatal disease in 1960s when it was known as a hyperacute fatal illness, it has now become one of the highest leukemias with highest cure rate, right? So if someone has leukemia, it's probably best to have ATML, which is very highly curable. So that's the journey, and you can see that in favorable these patients, what we give in these patients are just Atra and ATO, so it's a completely chemotherapy-free regimen, completely target therapy-based regimen, you could say. So that kind of therapy is possible, and with that very high cure rates are possible with ATML. So there could be some morphology variation. What about this case? Maybe Preeti can take this. Can you describe this cell for me, Preeti?
Yes, sir.
Yes.
It's a large cells with a moderate orbital cytoplasm,
Mhm.
fine chromatin, the 2 to 3 prominent nucleoli, sir. But
Okay.
there are less amount of granules, so it can be hypogranular variant of ATML, sir.
What about any other differentiation? Morphology, that's your only differentiation?
Nuclear indentions are seen, sir.
True. Now I'm asking you morphology, is this your only differentiation or anything else you will consider? Could there be any other entity that have kind of similar? I agree with you that this kind of cells look very, very suspicious for
No,
ATML.
it also some monocytic cells.
Correct. So
and
monocytic
monocytic
leukemias may have similar morphology, right? So how do you solve
Yes,
this
sir.
differential in a Friday night? So you are on a Saturday, let's say you are on call for Saturday night, and you have this morphology, and your differentials are hypogranular ATML versus acute monocytic leukemia. So the flow cytometrist tells you that go to hell. This is now 12 in the night. I will come in the morning, and then only I will do the flow. So at that point of time, can you do anything to differentiate these two?
kind of special state.
What special state?
Sir, NSC can do to look for monocytic lineage and
NSC can be positive in ATML. I agree with you. NSC is something that you can do, but they are actually positive in acute monocytic leukemia. Anything else? What is the first special that you do? Of course, MPO. So what do you expect in
Yeah.
What do you expect there? How did it help?
It will be positive in M3. human M3.
Strong positive in ATML.
Strong
When
positive.
we say strong positive, more than intensity, we mean that all these cells are positive in the same manner. Whereas in monocytic leukemia, what will happen?
Also only a little amount of cells will be positive.
Correct. So they can be either negative or they can be weak positive. Usually they will not show as intense, and all the cells will not stand for MPO. So that differential can be solved in special stain. Right. This is some special instances. I'm just showing you this for just for the purpose that you know that there could be varieties of ATMLs. This is a specific type of ATML which present the cells like this, which is monolobated. They do not usually have those figure of 8 type cells and associated dyspoisis. So you'll see some neutrophils like these, which have this bilobe nuclei, hypogranular neutrophil, and this kind of monolobated monomorphic promyelocyte. So this is typically seen in a variant of ATML known as ZBDB16 RARA. But just to take on the point that there could be variant types of ATML which has not PML but some other fusions, other genes fusing with RARA, and they can give you morphology like this. So you should be aware of all this morphology variations when you're dealing with ATML. Okay. So now let's start with our bonus case. Basically, this is the 14-year-old male now, and this is the morphology. I'll just go to the picture. So maybe who has not spoken yet? Everybody has spoken. So Tapan, maybe you can take this. How do you describe this morphology?
Sir, cells are large, almost more than 2-3 times the size of a small mature lymphocyte. The cytoplasm shows granulation and some speculation as well. Nucleus is large with increased density ratio, irregular nuclear contour, fine chromatin, few nucleoli, sir. And
So do all the cells look same to you, or there are some
also
different
variation in the shape of the nucleus.
Okay, there may be some variation, but about the myeloid maturational stage that we broadly think in
Okay.
mind. From that perspective, are they all same or there are maturation going on?
Well, appears same, sir.
Appear same, right? Again, some other picture. So what is your diagnosis? Give me diagnosis. This is something where you have to give diagnosis, right?
So again, maturation, I'd like to think of APML.
Yeah, of course you will think of APML. That's the only thing you will think of initially, right? So
Yes,
let's say you are in the on-call and you have let them know, and you did MPO, which was again nicely positive. So you let the oncology team know that this looks like an APML. You did flow also. I'm not going to detail. They had similar features like what you typically see in APML, right? So APML diagnosis confirmed. They have started with ATRA. Everything is good. But now the fish result comes. Okay. So you can see that this 2 green and 2 reds are completely separated. No fusion happening. So what is your diagnosis now?
Sir, again, RARA can be fused with any other gene other than PML, right?
So you think this is still APML, right?
Sir.
Although fish is negative. So before thinking variant RARA, there could be situations where fish is negative but PCR is positive. This can happen with many fusions. This is something known as cryptic fusions. Okay, so we go ahead and did our PCR as well. This you can see this is the positive control in a patient, in a known case, and this is our patient. So there is no band here. So what is the interpretation of PCR?
Negative.
Negative.
Negative
So
statement rather.
very good. So fish negative, PCR negative. Still stick to the diagnosis of APML, or you want to change?
So in the previous fish picture, the second image, it shows yellow.
Correct. So, very good that you have picked that up. So, this is a different type of fish. So, you see the figure A is a dual color fusion probe, right? Where one PML is red and RARA is green. But here, this is known as break apart probe. So, what it means is this is only for RARA. So, in normal situation, they will be fused. So, in break apart means if RARA is getting rearranged with something, this probes will break up, and then you will see separate red and green signal. So, in this case, the fact that you are seeing them fused, that means this is normal. There is no RARA rearrangement happening. Okay. So, I think Tapan, you told me that there could still be variants of RARA, right? But variants of RARA, you would have seen this RARA break apart being broken, right? It's not broken, it's intact. So, there is no RARA rearrangement. So, there is no PML RARA, there is no RARA rearrangement. So, would you revise your dimension now?
Yes, sir. But I'm not sure quite how to revise.
What about you, Sayanto? Still a PML, or you think the PML is not possible because you have proven that PML RARA is negative and RARA rearrangement is negative?
Not quite sure, sir.
Anyone else who wants to stick with your morphology, or you want to change based on the molecular result? Very fundamental point we are standing now.
So you can stick to the morphology.
Yeah,
So it is the case of ADPM.
stick to the morphology. People are saying stick to the morphology. So we are at our heart morphology, right? So we want to stick to our morphology. Very good. So then but you have to prove it, right? Because the oncologists will not believe. They will say, what are you saying? I've done all this and it's negative. So any other test that you can think of which can now help, like when all the PCR are really not being able to give the answer? So this is the moment of next generation sequencing, right? So we have to bring in the next generation sequencing to see if anything else we can see. So just before we go there, just to again the techniques we are not going to detail, but just to brief you about it a little bit. So this was Human Genome Project, which was covered in 2001. There's a long, huge history about it. You should read it. It's very, very exciting. It's how one of the greatest achievements of science was done using Sanger sequencing, if you can believe that. So in 2001, people used Sanger sequencing. There was a lot of story there. Like there was a government group in US which was trying to do. There was a private group, and how they came together finally, and how the private group was about to beat the government when Bill Clinton suddenly decided that no, we have to get these guys back in and we have to do it as a team. So all those fascinating histories there which you could read, which you should read actually. But this was the Human Genome Project that was covered in 2001 using Sanger sequencing. Right? Till that time, next generation sequencing was just coming up. And we knew that it was painstaking to complete the human genome using Sanger, but still people did that using billions of dollars at that point of time. But now we are in a situation where this is genome assembly. This is known as genome assembly. So now we are in a situation where we have full telomere to telomere genomes are complete. So except Y chromosome, for all human chromosomes now we know gapless sequences. So we know right from the start till end what is the sequence, and that this has been achieved only because of next generation sequencing, which includes long read sequencing, which is basically known as third generation sequencing, but predominantly short read next generation sequencing, right? So how this achievement was done is a really fascinating story of how next generation sequencing has impacted our life. And now actually we are going to a future where we will have pan genomes. So what it means is we know that my genome is not really completely same to your genome. Okay, there's always some variations happening. So far we have only one linear genome as a reference, as a human genome reference. But now people have realized that there are a lot of variations. So maybe we'll have a situation where, like in India, we'll have many states. From many states, we'll have different genomes and we'll combine them together. And when a patient comes from, for example, West Bengal, we will go back to the West Bengal reference genome. When someone comes from Delhi, we'll go back to Delhi reference genome, something like that. So that kind of future is also now we are envisioning. So if you think in broad term, like how sequencing has changed our life, so these are the common applications that you can think of. So one is this thing that I have spoken about, which is genome assembly. So like right from Human Genome Project to gapless human genome sequencing to now pan genome. So all these are genome assembly. Then there are applications like non-invasive prenatal testing, which you can do from the blood of the mother. You can know about the fetal DNA, and you could take important decisions on whether the child is suffering from genetic disease or not. In cancer, you could detect all the type of genetic mutations that's happening, and then you could use sequencer as counting devices, and you could do a gene expression, you could do ribosomal translation counting, you could do chromatin accessibility. So all those things can be done, right? So this is Sanger sequencing. This Shweta has discussed. I'm not going to details. So basically, again, this fluorescent level dyes are randomly getting incorporated into the elongating chain, and the chain is getting terminated, and you are based on that you're counting and you're diagnosing that. Okay, so this nucleotide is T, this nucleotide is G, and so on. So if I talk about next generation sequencing, you must have read a bit about it, right? Because you are attending a molecular class. So can anyone tell me like what is the basic difference in one word? How next generation sequencing is different than Sanger? Why is it next generation? What is so different in it? How does it allow us to be fast about it? So what is the basic difference of next generation versus Sanger sequencing?
So Sanger goes from one point linearly forwards.
Very good. And next generation?
Next gen, sir, a particular target means area, it will amplify, and then it will simultaneously read all the simultaneously read multiple copies of the same area.
Very good. So what is known as massive parallelization. So that means is you can do this sequencing in many DNAs together simultaneously. So therefore you could now, let's say I have 137 gene panel of active leukemia in my lab, in our lab, and all those, even within all those, we have many areas of DNA that we want to read, and all of them can be read simultaneously. So if to do that using Sanger would have been a real pain to do it, and it would have taken a lot of time, right? But with next gen sequencing you do it real fast. So we will just discuss a little bit about the principle on how it is achieved using the most dominating next generation sequencing technique in the market today, which is Illumina, who uses something known as sequencing by synthesis. So first in this stage, the critical thing is first to get a like a clonal amplification done. Okay, so how it is achieved this bridge amplification, where we have stretches of DNA that are tagged with adapters, as you can see here, these purple adapters, and then there are this in the flow cell of the instrument you have these areas where this adapter complementary sequences are there, and therefore your tagged DNA goes and bind them, and then the other part of the tagged DNA kind of folds on itself and gets bound to this other adapter here, and based on this sequencing happens. Now this is duplicated, and again they are straight, and again they will be duplicated, and this way we get a clonal amplification of this particular fragment. You can see the same thing here, how this bridge amplification takes place, and then we have clusters of DNA that we now need to sequence, and these clusters can be generated simultaneously for many regions of DNA, as we spoke about, what is known as massive parallelization. So different parts of DNA can be sequenced together. So now in the sequencing part, what is really interesting and what you should know and what is the basic difference with Sanger is, here also we have this fluorescent tagged deoxynucleotides, okay, and here also when they are incorporated into this growing chain, you can see here like a C is incorporated, here a G is incorporated. So at that point of time the chain elongation will stop because this fluorescent are tagged with this DNTP, and they cannot really further elongate. So this is stopped at this point of time. You then wash away the unbound nucleotides, and you then take an image. Because of this fluorescent, they will emit a fluorescence, and based on that you can decide whether it's C or G or T and so on. But how it is different from Sanger is the next step. So the next step, there is a step where this dye is cleaved, and now this sequencing can again go on. So what is known as reversible termination. So in Sanger the termination was permanent, and you had to then using electrophoresis, using this sequences you need to purify, and then you need to count manually. Whereas here you take this image and then the sequencing again goes on. So this reversible termination is the key which makes Illumina so successful, which makes them a $1 trillion company today. So that is how this sequencing thing is achieved. So I think enough on technique so far. So any question on this you could put in the chat box, but now we'll go on with the case. So in this case we did a modular RNA sequencing assay in our lab. So which is this RNA sequencing assay known as NARSIMA, which we have developed in our lab, and I'm not going to bore you with the chemistry of it, but just you should know that here the technique is designed in such a way that if you know one partner of the fusion gene, so for example if BCR-ABL, if you just know ABL1, we can detect any fusion partner of it. So you could detect BCR-ABL1, you could detect ETK6-ABL1, you could detect NUC1 for ABL1 just because you know only ABL1. So that is the technique that we use. So using that you can detect a lot of these different fusions. So in this case, I hope we all are at base with our case, which was a case of APML that the morphologist in us said, but in molecular so far it seems like negative, right? So in this case, we did RNA sequencing and it indeed picked up this fusion known as TBL1 XR1 RB. You do not need to know this, but just the fact that using NGS you can pick up fusions which are not possible to pick up using conventional technique. So usually we have RARA, like RAR alpha gene fusions, but sometimes rarely there can be RAR beta gene fusions, which gives rise to similar kind of picture. So the take home from this case is that sometimes morphology still is very important. So if you are confident on your morphology, especially on APML, you should stick to your diagnosis and you should do all these different sequence of tests to prove that you are dealing with them or not. And they have clinical importance as well. So this is known as APL like AML. This kind of this, like where we have fusions in either RAR beta or RAR gamma. So they have slightly worse prognosis as compared to RAR alpha APMLs, and their response rate to ATRA is also low. So therefore they not only help you in diagnosing, but also they will help you in prognosticating the patient. So what we have learned today from these two cases of APML, we know that APML is a morphologic diagnosis. Okay? The diagnosis of APL sits at the top of differential diagnosis of acute leukemia, and morphologic variations can exist. Sometimes, like in our first case, they can present with cytopenia. So you should be very, very meticulous when you're looking at peripheral blood. You should check the edges for those clues, those cells. They do express strong cytochemical MPO, and also this is something we didn't discuss. So it's okay to overcall this entity, right? So you're allowed to call something APML and then it turns out to be AML. That's fine, but you're not allowed to miss it at all. So you have to diagnose this entity. Sometimes even if you overcall, that's fine. And immunofluorescence is helpful, although they're not very specific. But look out for autofluorescence. Like in our case, CD34 seemed to be positive, whereas it actually was not. So controls are must in these cases. So that's what we learned from APML. But what we did learn from molecular perspective. So we saw CML that was characterized by BCR-ABL. We saw APML that was characterized by AML DANA. So demonstration of various fusions often establishes definitive diagnosis in hematologic neoplasm. So in those cases, fission RT-PCR both are must because PCR identifies transcript that allows for sensitive monitoring, and sequencing, like you see in our second and the third case, can help. RNA sequencing can help in challenging cases to find out rare fusions. And from this, we get a bigger point also, which is that most of the molecular technique in routine lab are targeted. Okay, so we do not usually do whole exome or whole genome type of stuff. So we are only looking at very targeted regions. So therefore, there is no answer. The answer is not negative. The answer is the negative in a particular context. So when I say PCR was negative, I should not say that it is negative for APML. It just means that the transcript that I have checked in my PCR, those are negative. So when I say fish is negative, it means that fish is negative for PML DARA at its sensitivity level. It can be less than that. Let's say if 1% disease is there, fish is not going to pick it up. So whenever you are doing a molecular test, you must know what is the specific question that you are asking and what is the sensitivity level you are looking at. Only then you can make an informed answer to it. And just to get around this problem, we might be looking at a future very soon in all where we will do unbiased sequencing in all leukemias, like whole transcriptome or whole genome. So maybe that kind of future is also not very far away, and where we will not really require all these targeted things. We will probably do a unbiased broad spectrum sequencing in all leukemias. So I think that was my last slide. So one more thing I would like to say. So today, the session, not only today, even the session next day, the way we have planned is that this is going to be very case based. So this is basically aimed at residents. So it's going to be very case based. This is not hardcore molecular techniques. So we have not discussed in great detail what are the different molecular techniques. So if you're interested in that, you could consider scanning this QR code and join our YouTube channel, where we recently had a molecular workshop where we had in details, we had discussed very different facets of techniques, variant interpretation, HGBS nomenclature, and so on. And you could consider watching those videos. So you could just scan this QR code. So with this, I would like to stop and maybe we can take some questions now. Should I want to add anything? I think, yeah.
So for variants like we saw in the last case,
Yes.
is genetic sequence the only method for diagnosis, or we can use anything else also? Like we said before, that RT-PCR can be helpful in variants diagnosis.
Yeah. So you have to know. So the problem with PCR is that you have to know exactly what you're targeting. So if your differential diagnosis is now 100 fusions, so for example, our case it was TBL1 XR1 RAR beta. The RAR beta can get fused to other genes as well. Like there could be runs on RAR beta. So then how many probes are you going to put? And these are extremely rare situations, right? So for this, are you going to order probes? Are you going to keep it in your lab where some case might come in once in 2 years or something like that? So and that too, where there could be 100 different fusions. So you have to put 100 probes together to find out the diagnosis. So for these reasons, sequencing is important because it does not need your prior knowledge of the fusion partner. You can detect that using one go, using a single panel. So for these cases, sequencing is important. Similar to if you take that example to, let's say, JAK2V617F, which is the canonical hotspot mutation in MPN. So we know that mutation that can be done by PCR. But what about DNMT3? What about WT1, which have mutations throughout their gene? So can you then go to design PCR for all those hotspots, which maybe hundreds or 200 or 53? So that's not possible, right? So in that case, you got to resort to sequencing, which is going to detect all these in one single test. And that is why there are massive parallelization. So you can detect 100 genes simultaneously at all their hotspots. So that is how sequencing is useful in these cases. Yeah.
Sir, I have some questions.
Yes. This is Uttapan, is it?
No, sir, I'm Shanta.
Yes, Shanta, go for it.
So one question is, can there be ordered without APML?
Well, you tell me. Can there be ordered with APML?
No, I think it can't be possible.
So AML with 821 doesn't show a rod. AML with Ramson fusion. MDS IP2 doesn't show a rods.
Okay.
So all of them do. Like this can be seen in myeloblasts. So many types of. In fact, if you look at the how you should approach acute leukemia on morphology, the top differential is ORROD. So only one thing that differentiates myeloblast from lymphoblast on morphology with absolute specificity is ORROD. Nothing else. Okay. So if you find ORROD, you know that you're dealing with myeloblast, right? So many types of AML, including some common ones like I said, AML with Ramson 21, AML with NPM1, MDS with IP2, so excess blast 2 that is. So all of them do show ORRODs in their cytoplasm. In APML, you see those faggots or you see collection of ORRODs. Those kind of things you more commonly see in APML, but still it's difficult to argue that those are absolutely specific for APML.
So my next question is, in the first slide there was a information FLT3 ITD ratio low.
Yeah.
How is this done, and why from that point can we think about NPM1 mutation?
Yeah, so again, see, FLT3 ITD is again a secondary mutation in AML. It can be seen in different types of AML, including APML. So in this case, that was present, but APML can show FLT3 ITD. In the next class, we plan to take on this issue further, like how do you detect NPM1 and FLT3 ITD and how that helps you. But again, remember, the FLT3 ITD is not specific for any genomic entity. So for example, if you have read in recent WHO or even WHO 2017, so you have seen an entity called AML with NPM1, right? You have seen an entity called AML with Ramson 21, but have you seen an entity called AML with FLT3 ITD?
No, sir.
Whereas FLT3 ITD is seen in 30% of AMLs. Maybe it is just less common than NPM1. Both are very common in AML. So why it is not a specific entity then? It's because it is a secondary mutation. It usually happens in many different types of AML. So although they are common, but they are not subtype defining. So FLT3 ITD will not make you diagnose a particular type of AML, but they are very helpful in some certain other perspective, which we'll discuss in next class.
And so another thing is, in the pictures of morphology, there was a cup-like projection in the myeloblasts, and cup-like projection is very specific to NPM1, isn't it?
It's
So
not.
was it there? Like was it in today's case, or have you seen the next next presentation's case?
No. From today's case, I'm talking about.
So first of all, these are not specific things. That is why we do not diagnose AML with NPM1 based on couple-like features. Do we diagnose that? Have you seen anyone diagnose AML with NPM1 based on couple-like features, right? So again, see, the problem is like Shweta says, in today's cases, we could argue that there are no specific couple-like things. There are more granularity in cytoplasm. There are some figure of 8 separate cells which we do not see in NPM1. So there are morphology clues, of course. But the larger point that we must emphasize is we should not diagnose these entities just based on morphology. There are some clues, of course, there are clues on which you can suspect that a certain entity is there, but do not diagnose them on morphology. You have to do that particular technique to establish them. We'll show you more example of couple-like feature and NPM1 in next class.
And so the last one is, can we call it an APL without PML RARA?
You tell me now, you have gone through the case 3. What do you think?
No, that RAR B, RAR gamma, that is the APL, PML RARA like.
What about this ZBT16 RARA? You have showed a variant RARA also, no? What about that? Is that APML or not?
Yes. So in the WHO, there are 3 criteria for diagnostic criteria there, and there is one criteria showing that there should be PML RARA. So I was asking.
No, but we have seen the cases today, right? So we know there are a lot of variant fusions that can happen with RARA. PML RARA is a distinct disease entity. Similarly, there can be other RARA fusions which will have very similar morphology on morphology. So see, whenever you diagnose an entity, what do you mean by an entity? If I ask you a very fundamental question, let's say you say something is AML, what do you mean by it? Is it a specific thing that you have seen in your microscope? Is it a specific therapy that the clinician will initiate? Or is it a specific behavior of those cells that will happen in the patient's body?
I think specific behavior.
Correct. So in a very fundamental space, it is a behavior that determines that those things. No? So from a clinician perspective, we only want to know whether this patient will behave in a specific fashion and whether I could treat them in a specific fashion. So there are many fusions of RARA which do respond to ATRA, which have a very similar DIC can also happen, which have a very similar clinical presentation. So therefore, all practical purpose PML. Whether you want to call them PML RARA, PML, something else you want to call them variant RARA, PML is your choice, but it's not going to matter in the clinical perspective, right? So now that with molecular techniques, we have many of these things coming. I'm happy that you're asking these granular questions. There are many different of these things will come up. So for this, I think we have to remember this thing very importantly, that it is ultimately the behavior that's going to determine whether or not we should level this patient in that way or not, right? And maybe whether it's a specific therapy or not. So those are the 2 things that we should keep in our mind when you're diagnosing anything. Sometimes it's easy to get lost into classification that people have given. So rather than that, we should think about that specific behavior and specific clinical implication of that.
Okay, sir.
Very
Sir,
good. Anybody has a list of questions? Anybody else has a list of questions?
Regarding the last slide, sir,
Yes.
But there is nuclear coagulation is also there. So can we make the differential diagnosis of acute lymphoblastic leukemia?
With that amount of granularity and with that MPU setting?
That I can see that granularity in the last slide. That diagnosis.
MPO? What about MPO? How will you defend
What about
MPO?
OR-ROD? What about MPU? Can any lymphoblast show OR-ROD? Like, I mean, Santo has told us about OR-RODs, right? Can any lymphoblast show OR-RODs?
No, that's lymphoblastic only, but I was confused with that.
So that is why we emphasize the point that 2 things. One is that you have to know the whole context. Okay. So one thing is clinical. That's okay. What is the patient presentation and all that. But overall morphology context is also very important. So you are looking at a picture where all the cells look similar. They have granules. They have OR-RODs. They are showing strong MPO. So are you then going to focus all your cytoplasmic calculations? I think this is the thing I was talking about. Sometimes pathologists will get carried away with one feature or one description that I've read in some book. Those things doesn't matter in larger picture. You have to think in the whole context, right? So you have got OR-RODs, you have got granulations, you have got strong MPO. So then the calculation doesn't mean anything, right?
That's clinically the age of the patient was also younger.
Sorry.
So clinically age of patient. So APML can occur in younger patient, no?
Typically occurs in younger female, no? Like they present with menorrhagia. There are younger females typically have APML.
I think there's a question in the chat saying, why do you see specifically in peripheral blood edges?
Yeah, so those are for cytopenic patient, right? So what I wanted to say is that in sometimes APML can present with cytopenia. So you should be very careful. You should be very meticulous when you're doing peripheral blood. So if you have seen peripheral blood, where do you usually find larger cells? Are they going to be well distributed throughout the smear, or they kind of stick to the edges, right? So many times they can be more concentrated on edges. So if you're looking at a very cytopenic picture, it might be helpful in looking at edges. You should use all sorts of tricks when you're looking at morphology.
It may young also mixed phenotype.
Sorry.
Mixed phenotype.
So how do you diagnose mixed phenotypic leukemia? Why do you have that thought in mind?
Because I thought that the cytoplasmic coagulation in the nucleus heterozygote.
How do you diagnose mixed phenotypic leukemia? That's the question again, right? How do you diagnose that? Is it based on morphology calculations?
Is morphological calculation specific for anything?
Correct.
Like morphological calculation can happen in anything. It is not specific for any entity per se. So why are you thinking that presence of calculation would be suggesting lymphoblastic lineage per se?
Correct. And also the diagnosis of MPAL is based on immunophenotyping, right? We have to see lineage specific markers whether they're based or not. It is never done on morphology. There is no morphology feature of MPAL that is specific to MPAL. You can never make a diagnosis of MPAL on morphology. That thing you should know. So that has to be an immunophenotypic call. It can never be made on morphology. So again, we have to be sure about the limitation of our techniques also, right? We might be morphology romantics in our heart, but you have to know at what point we have to stop interpreting. We shouldn't over-interpret findings. So like I've discussed on morphology, only specific thing that can tell you this is to some lineages or not. Or is specific for myeloid lineage. No other morphology feature, even granulation, no other morphology is specific for either myeloid or lymphoid lineage.
So regarding the last image, sir,
Yes.
the fact that whole transcriptomics and whole exome sequencing, so will it help to detect the broader chromosomal abnormalities? Is there any way which can
course.
use molecular test for chromosomal deletion and all large lesions?
There are many ways, Tapan. I'm very happy that you asked this question. So, there are many ways. So if you do whole genome or whole exome sequencing, you definitely will pick up so what you're asking is copy alteration, right? So what happens at chromosome level or even gene level. So that can be picked up using whole exome sequencing, whole genome sequencing. That can even be picked up using targeted panels. So for example, in our lab for all leukemias, in addition to this 137 gene that I spoke about, we do complete whole genome-based copy number changes in all cases. That can be done using sequencing-based techniques. There are newer things called long-read sequencing which are specifically useful for this chromosomal changes picking up. So in today's date, in all leukemia in Tata Memorial Hospital is diagnosed for copy number alteration along with mutation. So that can be very easily done using even panel-based targeted sequencing.
So in your opinion, sir, will molecular then means will the cytogenetic test will become done then after the molecular?
The class or is some kind of interview or some kind of thing that you come to.
No. Sorry, means your opinion.
That future, let's say that we are looking at that future that someday molecular would be replacing cytogenetic.
See, it's very easy to say that, but we need evidence to prove it, right? So I'm sure this kind of future will happen, but we need the evidence to show that. So maybe we and other groups will establish evidence, and then only we can say that. I hope there is no cytogenetics in the crowd. I'm getting extremely irritated by the discussion. But it's very good. I'm very happy that you guys are asking so many questions.
So someone
Why or? So I think this is Asteria. He or she asked that any leukemia diagnosis concept of PB results, cytogenetics, and immunophenotyping or genetic sequencing. So why do you put an or there? It's and, right? It's all the things together that makes the acute leukemia diagnosis. Having said that, are we looking at a future where a broad spectrum genetic sequencing could, in theory, replace all of this? Maybe yes. But again, like I said, that has to be shown and that has to be proven first. If that can be done in a cost-effective manner, if that can be done within an acceptable time frame for reporting. So those things need to be seen. But in today's time point, it's always and. There is no or in this thing. It's PB result, it's bone marrow morphology, it's cytogenetics, it's immunophenotyping, and it's sequencing. All together makes a diagnosis. And remember that today's data, like Shweta has shown you, it's not only diagnosing, right? You have to prognosticate. So if you diagnose an AML, are you going to stop at AML? You're going to re-stratify based on ALN. You're going to have to resort to MRD testing. You have to design MRD test result. So all these things you have to keep in mind.
For one more thing, I would like to emphasize, no matter what, morphology can never get replaced. You know, you can get BCR ABL in BLL also, you can get in CML also, and you can get in AMLs also. So how do you differentiate between these? Like, you would need morphology, you would need the clinical picture, everything. You have to integrate the findings, but you cannot replace morphology anytime.
Yeah.
That will never happen.
Sir, what is your opinion regarding the epigenetic test and all? Will that be helpful regarding diagnosis and prognostication?
See, information is always helpful, but you have to know that whether the cost and the resources that you will that you will use for it can be justified using the result that you're getting, right? So there's something, for example, in JNML, I think it is now shown, juvenile myelomonocytic leukemia I'm talking about. It is shown that the epigenetic patterns are important in predicting outcomes. So maybe in that space it is important. So in many things, this has to be evidence proof. So the point is that science go by anyone's opinion. It has to be data and evidence backed. So if somebody shows something that which is only possible using epigenetics, and then that can be done in clinics, we have to do that. But in today's day, epigenetics is not mandatory for leukemia diagnosis. We don't do it, and no routine lab involved does it.
It is going on in research level,
Yes.
but as of now, we are not having it in diagnostic.
Yes.
So it's actually wonderful interacting with you people, like the level of enthusiasm, and at the end of session also, you people are like, wanting to get your questions answered. That is really wonderful.
So I hope that coffee with Tapan and coffee with Santo finally ended, I guess. Or is there anything else, Tapan and Santo?
No, sir.
No, sir.
Great. So is there anything else we need to do, moderators, for the next session?
No, sir, nothing else. And then